1017, viral DNA may become part of the host chromosome. I'm going to do the best I can here. I haven't had a voice for a few days due to being ill. This material is not super important. You may have it in another class. I'm not going to focus on it on the test. So I would listen to it, but then I wouldn't spend a whole lot of time studying it for the exam. So a virus is essentially just genes in a box. It is you could almost think of it as a container that's made up of proteins that has genetic material inside of it. That genetic material can be DNA or RNA. The protein code I was talking about is called a capsid, and in some cases when it exits the host cell, it can actually take that host cell membrane with it, which then will make it have this membrane envelope, which helps it to avoid detection from the host immune system, because it can't tell that it's a foreign tissue with having its own cell's membranes wrapped around it. There are two types of reproductive cycles that viruses can go through, and one is called the lytic, and that's where the virus gets into the bacterial cell and replicates over and over and over again until the cell is so full it ruptures and releases thousands of viral particles, which will then infect neighboring cells. In the lysogenic cycle, the viral DNA is inserted into the host chromosome by a recombination. So the viral DNA is duplicated when the host cell is duplicated. So basically what it's saying is the viral DNA kind of hijacks the cell. It gets in there and it puts its DNA inside of the host cell. So whenever the host cell replicates, it's not only copying its own DNA, but it's also copying the viral DNA. This is what happens with HIV. So HIV will get in, it'll insert its information into the white blood cells DNA, and then when the white blood cell divides, now you have two cells that are carrying that virus, and then they divide, and then you have four, and it goes on and on and on, and soon all your white blood cells have this HIV virus inserted into it. And then for whatever reason, we haven't figured this out, but something then will signal the virus to go from that dormant phase hidden inside the host cell's DNA into an active phase, and then it will go into that lytic cycle and start pumping out viral viruses. So then at that point, that's when all the white blood cells end up breaking apart, and then the person who has that HIV virus has now just lost its immune system. So if we look at this picture, which didn't transfer in really well, we have where this little bacterial phage, it's a virus that infects bacterial cells, injects its DNA inside the cell, and then we go ahead and we make new viral particles, and then it ends up lysing the bacterial cell. So this is the lytic phase. In comparison, we have both phases here. So we have, again, that bacteria inserting its DNA. But in this case, over here, it ends up putting that DNA inside the host cell, and then the host cell divides, and then we have many, many, many cells that has that hidden virus inside of it. And then, again, something triggers that dormancy, and then we go to the lytic phase. So typical animal virus has a membranous outer envelope that has these proteins with sugars called glycoproteins, and that envelope will then help the virus enter and leave the host cell. So it's kind of, I always refer to it as the wolf in sheep's clothing. So it's kind of hiding underneath this envelope. So many animal viruses have RNA rather than DNA as their genetic material. And some of these viruses include the common cold, which is a rhinovirus, measles, mumps, polio, and AIDS, that use RNA versus DNA as their genetic material. So here is a diagram showing that in this case we have that membranous envelope. And what that membranous envelope is doing is allowing access by endocytosis. So literally the cell is taking in that viral particle because it's being told to by these receptors. We talked a little bit about receptor-mediated endocytosis, I believe in chapter 5. And so it takes it in, and then it will open up that vacuole, and then here we've got the RNA. Now the RNA will have a protein in there called a reverse transcriptase, and then it will make DNA, and then from the DNA we can make messenger RNAs. And then we have the host cell will replicate that mRNA just like it's its own mRNA, and then it will assemble what it needs to make that virus, and then it will exit by exocytosis, taking the membrane along with it. Now, when we look at the evolution, it looks like viruses that appear suddenly are new to medical scientists. We call them emerging viruses, and this includes AIDS, Ebola, West Nile, and SARS. There was a big SARS scare. Oh, it seems like yesterday, but it was probably about almost 10 years ago. It was primarily affected people in China and Canada. And of course, we deal here with West Nile virus with the stupid mosquitoes. And then, of course, this year we had the Ebola outbreak, and we're always dealing here with the AIDS virus. Which the AIDS virus really, I don't like how they put that, because AIDS is the symptom of the HIV. The virus itself is HIV. AIDS is the symptoms that you get from not having an immune system. That's more of, like, you could say, the symptomatic progression of that disease, not the actual virus. The virus is HIV. So 3 processes that contribute to the emergence, and one is mutation. Especially with RNA viruses, they do not have a proofreading mechanism, so they tend to mutate rapidly, and that's why we have so many colds out there. It's also the problem why we don't have a vaccine, or leads to not having a vaccine for HIV, because the virus keeps mutating so fast that the things that we normally would target using a vaccine, we can't keep up with it. We also, when you have different species that are in contact with one another, we find this in developing countries where they put all their livestock together, and you've got the duck and the pigs and all their animals that are kept together instead of separated, when they normally would not have been found together in the wild. And so what happens is viruses that normally wouldn't jump from one species to another are able to do that, and they may pick up some genes along the way, which allows them to then spread to humans. We also talk about the human population. With it being so crowded, it's easy for things to jump from person to person. And now that we are so internationally linked, and flights and ships are constantly going from country to country, we're pretty much it's a global problem today. As we found, it was very difficult to try to isolate the Ebola problem that was going on at the beginning of the school year. So AIDS, the acquired immunodeficiency syndrome, is, again, like I had already said, it's caused by the HIV virus. So it is RNA. You do have 2 copies of the RNA genome. One is the instructions, and the other one has instructions to make this protein called reverse transcriptase. Reverse transcriptase, because remember we talked about we can go from DNA to RNA by transcription, but we never talked about going the other way, and so that would be reverse transcriptase, and we have to have a special enzyme in order to do that, and that has to be brought along with the virus. So here's a picture of that lovely HIV virus, and you can see the envelope with the proteins that came from the surface of that white blood cell, the CD4, and we get into the GP120s. I mean, there's all kinds of information, but you don't really need to know that. And my dog has found another bone. Lovely. All right, so let me finish this slide before I take it from her again. And then here, this is the actual coat protein of the virus, and then we can see we have here the two RNA molecules, and of course here we have our enzymes coming along with us to reverse transcribe that information. So after HIV RNA is uncoated in the cytoplasm, we talked about the reverse transcriptase makes one DNA strand from RNA. And then the other reverse transcriptase is going to add the complementary DNA strand, and now we have a double-stranded viral DNA. It enters the nucleus, and then it becomes integrated. And we called the other one a prophage because it was from a what we call bacteriophage is the name of the virus that affects the bacteria. Well, this we're just going to call provirus. So now we have that provirus. Now it may stay in there and become part of that lysogenic pathway, or it may go lytic, in which then it will go from DNA, and it will go and make messenger RNA, which will make the viral particles to infect other cells. So here's a diagram of this, and you can see here we have our viral RNA. We have the reverse transcriptase. It's going to make a copy of DNA. We had to have that happen with the other piece as well. Now we have a complementary double-stranded molecule of DNA, and it will then splice itself into the host cell's chromosome. And now it will sit there and wait until a signal tells it to come out, and then it will go through its lytic phase, in which it will make the messenger RNA, which will make the instructions to make the virus, which will exit the cell. So the biggest problem with HIV is that you really cannot cure a virus. The only way to get rid of a virus is your immune system has to attack it and take care of it. This particular virus does exactly that. It attacks your white blood cells. So without having an immune system, you have nothing to fight this virus or any other pathogen, mold, anything in the air that comes into contact with your body. It basically decimates your immune system, and this is why this virus is so bad. So we have no tools in order to take care of this. We do have medicine that helps to slow this process down so that hopefully our immune system can take over and win. But for the most part, this is a death sentence. When you have HIV, this is something you will be fighting with for the rest of your life. Now, if that's not scary enough, there's actually some other things in there that we haven't really talked about, and they're called viroids and prions. Now, viroids are just very small circular strands of RNA, and they infect plants. And what they do is they replicate within the host cells without producing proteins and therefore interfere with plant growth. So basically what they're doing, and we talked about competitive and non-competitive inhibition. Well, this is kind of like that. It's competing with the plant's regular processes, so it slows down their growth. So it's more of just a big interference and slows things down. Now, a prion is a scary little molecule. Now, this is a protein. You could think of it as a zombie protein. So it is a protein that gets inside your body and will totally misfold other proteins. So it gets into your brain and it takes normal proteins and it folds them into this misfolded form and causes problems. It's basically causing chaos with the proteins in your brain. We call that mad cow disease. And what's really scary when you look at prions is A, we do not have a way to fight them, and B, we can't even kill them. It's like viruses, we can, use like Lysol and bleach and heat and all those things to sterilize things. None of those things work with prions. We have absolutely no way of killing prions. So now if we leave viruses and go to bacteria, there are three mechanisms to move genes from cell to cell. There's transformation, transduction, and conjugation. So we talked about how, binary fission, bacteria reproduce asexually, and so they're identical. But if they're identical, how do we get antibacterial resistance? How do we get different types of bacteria that don't respond to antibiotics? And how do we get some forms of E. coli that are harmless and others that are deadly? And this is how we're going to describe that here. So first of all, we have our normal bacterial chromosome. So this is just our regular instructions. But sometimes fragments or pieces of DNA, we can call it rogue DNA, can get into a cell, and we call this transduction. We also have another form where we can actually have a virus inject a fragment of DNA from one bacterial cell to the other. And that would be referred to as like a transformation. So we have all these different ways to mix things up here. Now, everybody does this, and even bacteria do this. They have sex. So there are different types. We call them positive, negatives, or not so much as male and female, but they can form this tube called a conjugation tube, and they can actually swap DNA through this conjugation tube. Kind of creepy thinking of bacteria have sex. In this picture, we're kind of showing how we have a piece of DNA here that's kind of been copied or cut out of the original, and how it's going to go over to another bacterial cell and cross over and then get integrated into a different bacterial cell. Now we have a recombinant chromosome. So like we said here, the ability to actually be a donor, the donor E. coli, to carry out conjugation, it's due, like I said, to this, we can all call it a positive or negative, or what we refer to as the F factor. So this F factor then, during conjugation, it's integrated into the bacterial chromosome, and then it allows it to start replicating that DNA, and then that DNA will then peel off and will go into the recipient cell. So this F factor serves as a leading end of the transferred DNA. So here you can see where we have the F factors integrated. We've got the donor, we've got the origin. That's where we're going to start our replication. We'll replicate, and then we're going to add it to a second, the recipient cell. The recipient cell will then integrate it into their DNA, and now it's recombinant. Besides making a copy, we can also have what are called plasmids. So plasmids are these little circular strands of DNA that are not part of the main genome, the main circle of the bacterial cell. They're these little additional pieces that are hanging out there. And many plasmids carry instructions for antibiotic resistant. So, which is really kind of scary, because you can make multiple copies of these plasmids, and then they put the conjugation tube, and then they swap, instead of swap and spit, they're swapping plasmids here. And this is going to be a problem, because then you can also have crossing over occurring, kind of like shuffling of the deck of genes on these plasmids. And this is what scientists are worried about. Eventually, we're going to end up with a bacterial cell that has multiple genes for antibiotic resistance, and we are seeing this in like MRSA. MRSA is a perfect example of this. MRSA is a bacterial cell that is resistant to many of the antibiotics that we use today. So here's just a picture of that. We've got the plasmid here. We're going to move on over, and we're going to make some copies, and we're going to add it over here. So MRSA, methicillin resistant Staphylococcus, oh, that was awful, aureus, or we could talk about, we've got Streptococcus fasciitis. That's another one. That's the flesh-eating bacteria. So all these things, what we're finding is that they're resistant to many of the antibiotics that we use today. So when we hear staph infections, people get really worried, because they tend to be the ones that are swapping things. We also have E. coli, which can carry certain types of toxins in those plasmids. And so you hear some people, they might have food poisoning and it may not be bad, and then you have other cases where many people are dying from those toxins in there. So we have to be really careful at some of the recombinant bacterial cells that are occurring today. So again, this information is more for your own information. It's really important to understand bacteria and viruses. If you take microbiology, you'll get much more into this. Not really what I'm going to be focusing on this test. So this test will be mostly focused on DNA replication, transcription, translation, meiosis, and mitosis. So focus on those main topics.